Okay, now in this today's session we will discuss the concept of, as we are going to start the parasympathomimetic. First, the question that usually come in your examination is, how does the acetylcholine get synthesized, how does it stores, and how does it metabolize inside the body. So before starting the parasympathomimetic, you have to make sure about the synthesis, storage and the destruction of acetylcholine, क्योंकि यूजुअल ये सेपरेट क्वेश्चन भी कई बार आता है. तो बेसिकली acetylcholine, as we know, this is the main neurotransmitter of cholinergic nervous system, or you can say the parasympathetic nervous system. So this is synthesized in the cholinergic nerves, or you can say the parasympathetic nerve. So here the red color line, this is indicating the presynaptic cholinergic neuron, right? And this is the postsynaptic cholinergic neuron that has their muscarinic and the nicotinic receptor, right? So acetylcholine, initially it will get synthesized with the help of ATP, acetate and coenzyme A. This is coenzyme A, right? So this is acetate activating reaction inside of the cholinergic neuron. So in the acetate activating reaction, the ATP and acetate in presence of coenzyme A form acetyl coenzyme A. This is acetate activating reaction. So in the acetate activating reaction, there will be the combining of ATP, acetate and coenzyme A, and they form acetyl coenzyme A and acetyl coenzyme A with the help of choline acetyl transferase, because acetyl coenzyme A further synthesized acetylcholine, but in this step there will be intake of choline. There will be acetyl coenzyme A with choline, and choline which is coming from outside of the cholinergic nerve ending with this green color transporter. This is sodium choline transporter, right? This is sodium choline cotransporter, allow the uptake of choline from outside of the cholinergic nerve end, and as it come inside, this choline is now influenced, or you can say the reacted with acetyl coenzyme A, and they both form acetylcholine and the enzyme become free. This coenzyme A become separated. Now this synthesized acetylcholine in the cholinergic nerve ending, it stores inside of this vesicle. This is synaptic vesicles of acetylcholine, and acetylcholine with the help of vesicular acetylcholine transporter. This is the vesicular acetylcholine transporter that allow the acetylcholine to come inside of the synaptic vesicle. Now this transporter helps the acetylcholine, this formed acetylcholine, to come inside of the synaptic vesicle, and whenever the impulse, or you can say the neurohumoral transmission will come, this synaptic vesicle will be exocytosis reaction will be there, and with the exocytosis the acetylcholine will be released, and the stored acetylcholine either will bind to their respective receptor, that can be muscarinic or that can be nicotinic, and produce their pharmacological response depending on the receptor where does it located, उसके आधार पे response देगा. And this acetylcholine in the synaptic cleft is exposed to a special enzyme called choline esterase, or you can say the acetylcholine esterase. Now this acetylcholine esterase degrade this acetylcholine into acetate and this choline, and the choline which is degraded, or you can say which is the byproduct of acetylcholine metabolism due to the exposure with acetylcholinesterase. This choline will be uptaken by or recycled by this sodium choline cotransporter, and again it is used in the synthesizing of acetylcholine again. And so this acetylcholinesterase is responsible for degrading the acetylcholine in the synaptic cleft. As we know, the acetylcholinesterase के दो टाइप होते हैं, and depending on the location and depending on the speed of hydrolysis, this acetylcholinesterase, which is present mainly on the synaptic cleft or cholinergic synaptic cleft, this is very fast, this produce a very fast hydrolysis of acetylcholine. Here I have mentioned that acetylcholinesterase, it is also called true cholinesterase. Another kind of cholinesterase is butyrcholinesterase, it is called pseudo cholinesterase. So acetylcholinesterase, or you can say the true cholinesterase, which is present in the synaptic cleft, here this is I am talking about, and this is present in the cholinergic synapse and in the red blood corpuscles. While the most of the drug of esterases category, they are metabolized by butyrcholinesterase. This butyrcholinesterase has the same work as acetylcholinesterase, but this is present on the plasma, mainly on the plasma, intestine and the liver. तो जो आप ड्रग लेते हैं एस्टर के फॉर्म में, वो मेटाबलाइज होती है थ्रू द ब्यूटाइलकोलिनेस्टरेज, और जबकि जो नेचुरल जो आपका एसिटाइलकोलिन है, दैट इज बीइंग हाइड्रोलाइज्ड बाय एसिटाइलकोलिन एस्टरेज. सो दिस इज द pathway of the synthesis of the acetylcholine, and this is the storage, that how does with the help of vesicular acetylcholine transporter it is come inside of the acetylcholine and it is stored in the ionic solution form, and how does it degraded with the help of acetylcholinesterase, that is of being two types, acetylcholinesterase and butyrcholinesterase. So this is all about the basic concept of synthesis, storage and destruction of acetylcholine. Thank you very much.